Have you ever wondered how bacteria can adapt so quickly to their environment, even acquiring resistance to antibiotics? It's a question that has puzzled scientists and medical professionals alike. The answer lies in the fascinating world of gene transfer, a process that allows bacteria to share genetic information. This process is a key player in the rapid evolution and adaptation of bacteria, enabling them to thrive in changing environments, survive in hostile conditions, and yes, even develop resistance to antibiotics. Gene transfer is not a single process, but rather a collection of mechanisms, specifically transformation, transduction, and conjugation. Each of these mechanisms has its unique modus operandi, but all aim at the same result: the transfer of genetic material from one bacterium to another, leading to genetic variation and evolution. Intriguing, isn't it? Well, let's dive into the mechanics behind this fascinating phenomenon. The first type of gene transfer we'll explore is transformation. In the grand scheme of life, transformation is a rather magical process, a bit like a scavenger hunt, but for bacteria. It's a process where bacteria can pick up pieces of DNA from their environment and incorporate them into their own genome. It's as if they're picking up a recipe from a cookbook left lying around and adding it to their own repertoire. Let's delve a bit deeper into this fascinating process. Imagine a bacterial cell in an environment where another bacterial cell has died and burst open, spilling its DNA into the surrounding milieu. This is where the transformation process begins. The first step is the death and lysis of a donor cell. Picture a bacterial cell that has reached the end of its life cycle. As it dies, it bursts open, or lyses, and its genetic material, the DNA, is released into the surrounding environment. Next, we come to the uptake of this free DNA by a recipient cell. Now, not all bacteria can take up this foreign DNA, only those that are competent, a state that is often induced by environmental conditions such as starvation. When a competent bacterium comes across this free DNA, it attaches to it and reels it in, much like a fisherman reeling in a catch. Finally, we arrive at the integration of this DNA into the recipient's genome. Once inside the recipient cell, the foreign DNA is integrated into the bacterium's own DNA. This can happen through a process called homologous recombination, where sections of DNA that have similar sequences line up and swap places. This way, the recipient bacterium has now gained a new piece of genetic material, which it can use to its advantage. So in essence, transformation is a bit like a scavenger hunt, with bacteria picking up useful bits of DNA from their surroundings. They're not just surviving, they're adapting, evolving, and growing smarter with every piece of genetic material they incorporate. Now, isn't that a mind-blowing thought? Next up, we have transduction, another fascinating method of gene transfer. Now, this isn't your typical passing of notes in class, oh no. This involves a bacteriophage, which is basically a virus that infects bacteria, acting as the courier. Let's break it down, shall we? Transduction begins when a bacteriophage gets cozy with a bacterium, the donor. Unlike in transformation, where the bacterium willingly gives up its DNA, here, the bacterium doesn't have much of a say. The bacteriophage infects the donor bacterium, and during this process it becomes a vehicle for the bacterium's DNA. Here's where it gets interesting. The bacteriophage, now carrying the bacterial DNA, takes off and sets its course for the next bacterium, the recipient. It's like a microscopic version of a mail carrier delivering a letter, only this letter can rewrite the recipient's genetic code. When the bacteriophage reaches the recipient bacterium, it infects it just as it did the donor, but this time, it brings along a little surprise, the donor's DNA. The bacteriophage injects the bacterial DNA into the recipient bacterium, and voila! The recipient bacterium now has new genetic material. It's important to note that this isn't a conscious process. The bacteriophage isn't intentionally playing matchmaker for bacterial DNA, it's just a consequence of the bacteriophage's life cycle. This method might seem indirect, but it's incredibly effective. It's one of the ways bacteria can gain new traits quickly, which helps them adapt to changing environments. In a nutshell, transduction is a process of gene transfer where a bacteriophage acts as the middleman, carrying DNA from one bacterium to another. It's a remarkable example of nature's ingenuity, displaying how even microscopic entities can have a significant impact on life as we know it. So, transduction is essentially bacterial gene transfer with a viral middleman. It's like nature's own version of a postal service, delivering genetic packages from one bacterial address to another. Last but not least, let's explore conjugation, a direct method of gene transfer. It's a fascinating process, really. Picture this. Two bacteria living in the same environment come into direct contact. This isn't just a casual bump or graze. This is a planned and purposeful meeting. It's as if one bacterium says to the other, "Hey, I've got some interesting genetic material. Would you like some?" And the other bacterium, ever curious and always looking to diversify its genome, says, "Sure, why not?" The process begins with the formation of a structure called a pilus. This pilus is like a bridge or a tunnel extending from the donor bacterium to the recipient. It's through this pilus that the genetic material, the DNA, will travel. But it's not just any DNA that gets transferred. It's a special kind of DNA called plasmid DNA. Plasmids are small circular pieces of DNA that exist separately from the chromosomal DNA in bacteria. They often carry genes that can give the bacterium an advantage, like resistance to antibiotics. So the donor bacterium replicates its plasmid DNA and pushes it through the pilus. It's like sending a coded message through a secret tunnel. On the other side, the recipient bacterium is ready and waiting. It receives the plasmid DNA and incorporates it into its own genome. This is a fantastic way for bacteria to share beneficial traits and rapidly adapt to their environment. It's a form of horizontal gene transfer, meaning that it happens between organisms of the same generation. And it's not just limited to bacteria. Conjugation can also occur in archaea and some types of yeast. So there you have it. Conjugation, a biological process that's part handshake, part secret message exchange, and all about survival and adaptation. Next time you think about bacteria, remember this intricate and fascinating process that they use to share genetic material and adapt to their environment. In a way, conjugation is like a bacterial handshake, but with the exchange of genetic material. So, we've learned about 3 incredible ways bacteria can share their genetic information. It's a fascinating world, isn't it? One that is both microscopic and magnanimous, with these tiny life forms using ingenious methods to ensure their survival. Let's take a moment to recap what we've discovered. First, we dove into the process of transformation. This is the remarkable ability of bacteria to pick up stray pieces of DNA from their environment and incorporate them into their own genetic material. It's like picking up a book you found on the street and adding its contents to your own knowledge base. But it's not just about adding new information, it's about adaptation, survival, and evolution. By incorporating new genetic material, bacteria can develop new traits, resist antibiotics, and even repair damaged DNA. Next, we explored the world of transduction, a process that could be likened to a sci-fi movie plot. Here, bacteriophages, or viruses that infect bacteria, play the role of interstellar couriers. These bacteriophages accidentally pick up bacterial DNA during one infection and deliver it to the next bacterial cell they invade. It's a game of chance, one that can result in significant genetic changes for the recipient bacteria. It's like receiving a parcel meant for your neighbor and finding inside a tool that can help you survive a crisis. Then, we uncovered the secrets of conjugation, the most direct form of gene transfer among bacteria. Imagine 2 bacteria, connected by a bridge, exchanging genetic information like whispers in the night. This is conjugation, a process that allows bacteria to share plasmids, small circular pieces of DNA, containing genes that can provide survival advantages, like antibiotic resistance. Each of these processes, transformation, transduction, and conjugation, play a crucial role in bacterial adaptation and evolution. They enable bacteria to acquire new traits, resist threats, and survive in changing environments. They are the tools of bacterial innovation, the mechanisms that drive the evolution of these microorganisms. Looking at this, we realize that bacteria aren't just simple, single-celled organisms. They are complex, adaptive, and innovative. They have developed remarkable ways to share their genetic secrets, to adapt, evolve, and survive. And that's something we can't help but admire. So the next time you marvel at the adaptability of bacteria, remember these ingenious methods they use to share their genetic secrets. It's a testament to the resilience of life, to the power of evolution, and to the intricate beauty of our natural world. And remember, this is just the tip of the iceberg. There's a whole world of scientific wonders waiting to be explored, so stick around, join us on this journey of discovery. Subscribe for more because you won't find me later.